We're now going to turn our attention to the B subject topics for IB Biology, and this is focused on form and function. And in this video, we'll begin to introduce the particular topics of carbohydrates and lipids. And in this first video, we're going to look exclusively at the examples of carbohydrates and what makes up carbohydrates and their particular functions. Before we get into the specifics of carbohydrates, it's important to understand the difference between something that's organic and inorganic. We're not talking about produce organics. We're talking about, in terms of organic, compounds that contain carbon. And so these would be things that are found in living organisms. Inorganic would be examples of compounds that don't contain carbon, with a few exceptions of carbon dioxide, carbonates, and hydrogen carbonates. And so typically, anything that involves carbon that we're going to talk about would be considered organic. And so now let's discuss what is carbon, the carbon atom. It's the 15th most abundant element. It's extremely essential to life. One of the main reasons that it's essential to life is because it is able to form covalent bonds by sharing electrons, and as a result of that, it's able to form actually 4 different bonds. Its ability to form covalent bonds is caused by the ability to share electrons between 2 adjacent atoms, in which the positive is attracted to the negative, and as a result of this, it creates a very strong type of bond, this covalent bond, and it allows for very stable molecules. And that's really essential to living things, is the ability to form lots of different types of stable molecules. And so it can form not only single bonds, or it can also form double bonds. So it could form 4 single bonds or 2 double bonds. And as a result of this, it makes up some major classes of molecules called macromolecules. And this would include carbohydrates, lipids, proteins. Those 3 you're probably familiar with as food and nutrition labels. And the 4th would be nucleic acids, which we've already discussed as part of DNA. The form of these different molecules can come in a couple of different types. It can form chains. Typically, we see this in fatty acids, lipids, rings. Glucose would be an example of this, or chains of rings, which would be found in starch, like what we find in potatoes. It's very tasty. Bonds of carbon atoms spread out as much as possible, and so typically when they're forming shapes, they form a tetrahedral shape. Because of this, they spread out as far as possible, and the chain is not always straight. It's oftentimes in a zigzag shape, and we'll look at this a little bit more specifically when we begin looking at some examples of polysaccharides shortly. Before we get into specific examples, we need to better understand how molecules made of carbon can be put together. And the first part of being able to do that is the ability to define something called a monomer, and that is a single subunit of a macromolecule, so a carb, lipid, a protein, or nucleic acid. A single unit, a building block, if you will, of each of those would be called a monomer, and a polymer then would be poly meaning many, and be many of those monomers put together. It's a whole chain of those subunits put together. And so within our macromolecules and organisms, there are different types of polymers. Polysaccharides, poly meaning many, saccharide meaning sugar, and so a polysaccharide would be a type of carbohydrates. Polypeptides would be proteins, and then nucleic acids. The monomers of each type of these molecules are linked together into a polymer by a process called a condensation reaction. And a condensation reaction is when 2 molecules are linked together, and as a result of that, 1 small molecule is released. Oftentimes that's water. And so anytime a condensation reaction is releasing water, we call that dehydration synthesis. And I always remember this as, if I'm dehydrated, I've lost water, and in this situation, these 2 molecules are being connected together by the loss of water. This process requires some energy to be able to facilitate, and that energy that's necessary is provided by ATP. The reactions that form polysaccharides or polypeptides or nucleic acids occur by the removal of specifically a hydroxyl group, an OH, from one molecule, and a hydrogen atom from the other molecule. And so we've got OH from one, H from the other. That gets removed, H2O, water, and then it creates a bond between those 2 different molecules. The opposite of this is called a hydrolysis reaction, and hydro means water, lys means to break, and so hydrolysis means that we're using water to break apart 2 molecules, and this would be the deconstruction of a polymer into monomers or into separate components. When you eat food, these reactions take place. Our body and the acids that our body produces in the stomach and the intestines helps to break apart food during the process of digestion. And so this would be the splitting of polymers into monomers so that they can be used for energy and for the production of ATP. It's essentially the opposite of condensation, in which water is used to break apart the molecules. OH, hydroxyl, goes to one molecule, and H goes to the other molecule in order to separate them. So this image provides a good clarification of this process, where we can see specifically two glucose molecules being put together by the process of condensation to form maltose and water, and the opposite for hydrolysis, in which the maltose is being broken apart with the addition of water into two separate individual glucose monomers. So now that we have an understanding of the different components and building blocks of monosaccharides and carbon-based molecules, we can begin to look at some specific examples. And we'll start with what we call a monosaccharide. And mono means one, and saccharide, as we have established already, means sugar. And so a monosaccharide means one sugar. And some examples of this would be the molecules of glucose, fructose, and galactose, the three monosaccharide sugars. They have typically 3 to 7 carbon atoms within monosaccharides. Glucose is the example that we're going to look at a little bit more specifically, and you maybe have heard of glucose before. It's essential to the ability to be able to produce ATP through the process of respiration, aerobic respiration. We'll discuss a little bit later on in the course. Glucose, as a molecule, is a 6-carbon sugar. Its molecular formula is C6H12O6, meaning it has 6 carbon, 12 hydrogen, and 6 oxygen. It's soluble, relatively small, and it's easily transported. It can also be transported in the bloodstream. It's very chemically stable. It's a very stable molecule. It can be converted to glycogen or starch for storage. We'll talk about both of those in just a moment. It can be combined to form polymers by a linkage between the first and fourth carbon on the molecule. And so within the chain, within the ring of glucose, it has 5 carbons, and then a 6th carbon is attached to carbon number 5. By combining multiple glucose molecules at carbon number 1 on one of the molecules and carbon number 4 on a second molecule, we can make cellulose or glycogen, these polymers, polysaccharides. And when glucose is oxidized through aerobic respiration, it yields energy. And specifically, it's 4 calories per gram for carbohydrates that's yielded. We can also see here in our images some examples of glucose as a 6-carbon sugar, as well as ribose as a pentose sugar, meaning it has 5 carbons in its ring. A disaccharide is our next step in the progression from monosaccharide. Disaccharide means 2 sugars put together. Can also be used as a source of fuel, can be converted to other molecules or combined into polymers. The disaccharide that you're probably most familiar with is lactose, the sugar that we find in dairy products. Sucrose is an example, represents what we see in table sugar. If you go to the store and buy sugar, you're buying sucrose. And maltose is a natural sweetener. And each of these are 2 of those monosaccharides put together and combined to be able to make the disaccharide. So that leads us to polysaccharides, and polysaccharides means many sugars put together. Some good examples of this would be starch in plants. It's for storage of energy within plants, and for animal energy storage, glycogen would be the other example. Both of these use large numbers of alpha glucose molecules. We'll talk about that in a moment. 2 different types for starch. Amylose would be an unbranched chain of alpha glucose with linkages at the 1,4 carbons that we talked about on glucose. So it's a whole bunch of glucose put together, and those linkages occur at carbons 1 and number 4. Amylopectin would be another example of starch, and this is the same in that it's an unbranched chain of alpha glucose, but there are some bonds that occur at carbons number 1 and number 6, so a little bit different, and this makes the molecule branched as opposed to just a chain in amylose. Glycogen is the energy storage that we find in animals, and this is similar to amylopectin, but it has more branching. So similar, but has a little bit more branching. Both of these, whether it's starch or glycogen, they act as an energy bank. And so what that means is that it's a whole bunch of glucose molecules put together, and to acquire energy, individual glucose molecules can be removed to gain energy, or if there's an excess amount of energy, they can be added to or deposited through condensation reactions, or hydrolysis reactions would remove them. And this helps to facilitate the mobilizing and use of energy stores. And so it's like an energy bank that you have access to that you can move or add energy to as needed. Both of these are really large molecules. They're thousands to hundreds of thousands of glucose molecules long. They're much less soluble than glucose, and so this allows them to be easily stored. Lots of energy can be stored within them, and because they're not soluble, it's also doesn't cause the cell to intake lots of water by osmosis, and so they have low osmotic concentration. So it's not going to impact how much water the cell takes in because they're not very soluble. Additionally, the size of the molecule, although they have a large molecular mass, the branching takes up a lot less space, so they're compact for how large of a molecule that they are. Not only can polysaccharides provide energy storage, but they can also be used for structures. And this is primarily found in plants. Cellulose is made of glucose, and it is what helps to make up the cell wall in plants. The cellulose, it uses beta glucose rather than alpha glucose, and it creates an unbranched chain. It can contain about 10,000 glucose molecules. And in order for the bonding to occur between the beta glucose at 1,4 carbons, the glucose molecules must be inverted. So they're inverted to one another in relation to the previous molecule so that it can be this continuous unbranched chain. What this does is it creates a straight chain, and the formation of the glucoses are arranged in a parallel function. And these can be bundled together to create microfibrils. And that forms the basis of the plant cell walls, all of these put together. And as a result of this, it has really high tensile strength. It's really strong due to these covalent bonds, as well as the number of molecules and cross links between hydrogen bonds. And what this does as a result is it prevents the plant from bursting. And so within plant cells, as we've looked at under the microscopes, the cell wall is built of these, and it helps as the cells take in water. It helps them to prevent from being able to burst, and it creates a really strong exterior for each individual cell because of this cellulose structure. Our last example to look at here is what's called a glycoprotein. And yes, this is a protein. It doesn't necessarily fit with carbohydrates. However, it's a protein that has a carbohydrate attached to it. And it's usually in the form of an oligosaccharide, which is a short chain of monosaccharides that are linked together by a glycosidic bonds. Typically with these, it's found in the plasma membrane of animal cells, and the carbohydrate portion is at least lies outside of the cell membrane. And what this does is it allows other cells to recognize the particular type of cell because of this carbohydrate that's attached. It helps with organization of tissue as well as identification of foreign objects. And a good example of this can be found with blood types, specifically the ABO blood types, ABO glycoproteins. They're found in red blood cells, and they serve as a good example. Every person has a particular blood type. You may know your own. A and B types reject one another. A and B accept any, and O is the universal donor in that it has the same structure as A and B with one monosaccharide less, so it's not recognized as a foreign object. And so this would be a good example of how a glycoprotein could be used to recognize foreign objects, because if somebody that is blood type A is given blood type B, then the body rejects it as a foreign object. And so as we get into membranes, we'll look at glycoproteins a little bit more in a future unit. And so this concludes our discussion of carbohydrates. Next, we'll turn our attention to the macromolecule called proteins.